children, कैसे हैं आप सब? Now we continue with the last part of this chapter, the fundamental unit of life. So we have discussed cell structure, plant cell, animal cell, cell organelles, and the topic left is diffusion, osmosis. What are diffusion, osmosis, and then isotonic, hypertonic, hypotonic solution. Diffusion is the movement of particles from an area of high concentration to low concentration. Its effect is to equalize concentration throughout the medium. Simple example of diffusion is when we open a bottle of perfume, in one corner of room we can smell it from other corner. It's only diffusion. Then diffusion of gases, carbon dioxide and oxygen, occur in cells, in tissues. Water also moves through diffusion, but when water moves through a semi-permeable membrane, it is called osmosis. So osmosis is special type of diffusion where movement of solvent, or you can say movement of water molecules, occur across a semi-permeable membrane from a dilute solution to concentrated solution. Or we can say movement of water from the region of high water concentration to the region of low water concentration through a semi-permeable membrane or selectively permeable membrane. It is called osmosis. Now, we have discussed when we were doing the functions of plasma membrane that plasma membrane is semi-permeable. It allows only certain molecules to pass through it. So, here is the importance of semi-permeable membrane, or it is also called selectively permeable membrane. That is why in the cells either oxygen diffuses or carbon dioxide diffuses or water comes in or go out. The basic difference between the diffusion and osmosis is that in osmosis there is movement through a semi-permeable membrane. In the diffusion, there may not be any membrane. You can say osmosis is also a diffusion, but every diffusion is not a osmosis. When in the diffusion, between the two parts, there is a semi-permeable membrane, then it is the osmosis. Now next, we come to the isotonic, hypertonic and hypotonic solution. So what is hypotonic solution? Like this is a hypotonic solution. If medium surrounding the cell, so this is a cell, this yellow portion is a cell, and medium, this bluish is the medium. If a medium surrounding a cell has higher water concentration than the cell, it means outside solution is dilute. See, these are given, these dots they represent the solute molecule. You see in the cell, ये cell है अंदर, इसमें कितने सारे 6 molecules दिखाए हैं और इसमें सिर्फ एक molecule है. So outer solution is dilute as compared to inside the cell. So this is called hypotonic solution. Outer solution is called hypotonic solution. In this case, what will happen? Water will move from outside to inside the cell, or you can say swelling of cell will occur. Then isotonic solution. Here you see that medium has same concentration as the cell. You are shown here 6 dots, again here 6 dots, means solute concentration is same. So when outer solution has same concentration as the cell, then movement of water will occur, but it will occur on both the side, inside to outside, outside to inside equally, but there is no net movement of water molecules, and this outer solution is called isotonic. Tonic means concentration, iso means same. So it has same concentration, and so no net movement of water occurs. Hypertonic. Hyper means higher. Here you see, outside the cell there are so many solute molecules, and inside the cell again 6 dots they are shown. So here the outer medium has higher solute concentration as compared to inside the cell. The water moves out of the cell, or in this case the cell shrinks. Now in the next slide you see the effect of this hypertonic, isotonic and hypotonic solution. Here in the hypertonic solution the water moves out. Water is moving out and the cell shrinks. If it is a plant cell, we say it is plasmolyzed because there is a cell wall around this cell. So protoplasm shrink, cell wall की shape ज़्यादा change नहीं होती है, and this is known as plasmolysis in the plant cell. अगर वो animal cell होगा तो पूरा का पूरा cell ही shrink हो जाएगा. But इसका मतलब ये नहीं है कि पानी अंदर नहीं आ रहा. It comes inside, go outside, but it moves more outside as compared to the inward movement. इसलिए वो shrink हो जाता. Here it is an isotonic solution moving out, in equally because the concentration is same. Flaccid cell we say. And this is the hypotonic solution. Here we see where the outer solution is dilute. Dilute means that outside there is more water. Water is coming into the cell. The cell is becoming turgid. Simply जैसे हम देखते हैं, हम एक रेजिन को जब हम पानी, रेजिन होता है किशमिश, अब हम उसको पानी में डुबो देते हैं तो एकदम उसकी swelling हो जाती है. So that is the due to the hypotonic or solution. So ये हमारा एक जो टॉपिक रह गया था. इसके बाद हम कुछ एक टेक्स्ट बुक के क्वेश्चंस डिस्कस करेंगे. यू हैव ऑलरेडी गिवन सम क्वेश्चंस. I think you must have done that difference between the prokaryotic and eukaryotic cell, diagram of plant cell, diagram of animal cell, and then differences between plant and animal cell. Then you have to do the functions of all organelles. This you have already done, and again show your pics in the group. And next a few questions, some thinking type of question. Differentiate between diffusion and osmosis. I think this you can easily do. In osmosis there is selectively permeable membrane between the two regions. In diffusion there is no selectively permeable membrane. Diffusion occur in the gases, is liquids, but the term osmosis we usually use for the water molecule. Another, an objective type question. This is not in your textbook. On the basis of your understanding of movement of molecules, write osmosis and diffusions in the giving example. The student sitting next to just came from gym class and forgot to shower, and you can tell. तो आपके पास कोई बच्चा आके बैठ रहा है, जिम से आया है, यानी कि बहुत सारी स्वेटिंग है उसमें, तो आपने बताना है कि उसको यहाँ पे ऑस्मोसिस है या डिफ्यूजन है. सो think and then write in your notebooks. Then another, oxygen molecules move from the air sacs in the lungs across the cell membrane into the blood. ऑक्सीजन मॉलिक्यूल्स एयर सैक्स लंग्स के बीच में हमारे ब्लड में जाती है, तो वो कौन सा प्रोसेस होगा? डिफ्यूजन या ऑस्मोसिस. द थर्ड वन इज रॉबर्ट स्प्रेज वाटर ऑन द वेजीज इन द प्रोड्यूस सेक्शन टू प्लम्प देम अप. वेजिटेबल्स के ऊपर वाटर स्प्रे किया जाता है टू प्लम्प देम अप, जस्ट टू वो थोड़ा फूल जाते हैं, फ्रेशन लगते हैं. तो आपने ये प्रोसेस का नाम बताना है कि यहाँ पे ऑस्मोसिस है या डिफ्यूजन है. नाउ देन few other question given in the textbook, that is the amoeba, diagram of the amoeba. Then this is again a textbook question. This is a slightly typical question, and it is also your activity. So I want to discuss this. Take 4 peeled potato halves and squeeze each one out to make potato cups. One of these potato cups should be made from a boiled potato. Put each potato cup in a trough containing water. So I'm just giving you the example to show that how these potato cups are made. See, this is a potato. Peel it from outside. Make a cavity inside the potato, and this membrane, it should be thin. There should not be any hole. Make a cavity. Then we place this in a container containing pure water. So just we take 4 such potato cups. 3 with the raw potato, 1 with the boiled potato. Just understand the question carefully. Keep cup A empty. Put 1 teaspoon sugar in cup B. Put 1 teaspoon salt in cup C. Put 1 teaspoon sugar in the boiled potato cup D. Keep these for 2 hours. Then observe the 4 potatoes cups and answer the following. This you can easily do at home. In this cavity, you add small amount of sugar or salt. Then keep this for 2 hours. You will see that water comes inside the cavity. You have not poured any water, but water comes inside the cavity. Now the reasons are, this is your textbook question. I have shown with the diagram also. See, this is the A, empty, B with the sugar, C with the salt, and D with the boiled potato. Keep cup A empty. Why water gathers in the hollowed portion of B and C? The first question is, explain why water gathers in hollowed portion of B and C. So here you see that water comes only in B and C. जो आप घर में भी चीनी डाल के रखोगे या सॉल्ट डाल के रखोगे, उसमें पानी अंदर आएगा. ना तो A में खाली वाले में आएगा, ना ही बॉइल्ड वाले में आएगा. तो ये दोनों में पानी क्यों आया? ड्यू टू द ऑस्मोसिस. यहाँ पे क्या है? प्योर वाटर. ठीक है, कोई सल्यूट नहीं है इसके, ना हमने सॉल्ट डाला ना शुगर डाला. यहाँ पे क्या है? शुगर. सो वाटर इज हियर इन द हायर कंसंट्रेशन. यहाँ पे पानी है ही नहीं है, तो वाटर मूव्स थ्रू द सेमी परमिएबल मेम्ब्रेन. सो दिस पोर्शन इट हैज मेनी सेल्स. पोटैटो एक सेल नहीं है, बहुत सारे सेल्स हैं. तो इन सब की मेम्ब्रेन से क्रॉस होके पानी अंदर चला जाता है. दिस इज व्हाट इज ऑस्मोसिस. हियर आल्सो द वाटर एंटर्स थ्रू द ऑस्मोसिस. नाउ कम टू द नेक्स्ट क्वेश्चन. व्हाई इज पोटैटो ए नेसेसरी फॉर दिस एक्सपेरिमेंट? अब पोटैटो ए को रखने की क्या जरूरत है? खाली पोटैटो रखने की क्या जरूरत है? Whenever we do our experiment, we always compare results. This is a control experiment. For comparison of our result, भाई अगर हम इसको नहीं रखेंगे तो हमें क्या पता खाली पोटैटो में भी पानी अंदर जाता हो। तो इसको देखने के लिए भी हाँ शुगर होगी, सॉल्ट होगा, तभी पानी अंदर जाएगा, खाली में नहीं जाएगा। वी हैव टू यूज अ कंट्रोल एक्सपेरिमेंट। वी ऑल जब भी हम कोई भी छोटा सा भी एक्सपेरिमेंट करते हैं, एक्टिविटी करते हैं, वी ऑलवेज यूज अ कंट्रोल जस्ट टू कंपेयर अवर रिजल्ट्स। सो ए जो है इट इज यूज्ड एज अ कंट्रोल एक्सपेरिमेंट टू कंपेयर आवर रिजल्ट. Then the last question here is explain why water doesn't gather in the hollowed out portions of A and D. अब water ना A में आया ना D में आया. This is D. तो water ना A में आया ना D में आया. A में क्यों नहीं आया? बिकॉज देर वॉज नो डिफरेंस ऑफ कंसंट्रेशन. यहाँ पे हमने कुछ रखा ही नहीं था तो ऑस्मोसिस नहीं हुई, नहीं आया. डी में क्यों नहीं आया? यहाँ तो हमने शुगर रखी थी तब भी क्यों नहीं आया? यहाँ हमने शुगर रखे, सॉल्ट रखे, तब भी नहीं आया भाई. क्योंकि यहाँ हमने लिया बॉइल्ड पोटैटो, मीन्स सेल्स आर डेड. जब सेल डेड हो गए तो प्लाज्मा मेम्ब्रेन हैज लॉस्ट इट्स प्रॉपर्टी ऑफ सेमी परमेबिलिटी. अब उसमें से वाटर ट्रांसपोर्ट नहीं होगा. सो ऑस्मोसिस दैट डजंट अकर वो लिविंग सेल में ही हो रही है. सो दिस क्वेश्चन of your textbook I especially want to discuss. Others I think you can do, and then diagrams of prokaryotic, eukaryotic cells as and diagram of plant cell, animal cell, difference between the two you will do in your notebooks. And another activity is to prepare the onion peel slide that we will do in the our labs. But just I will tell you a little bit कि हम इसको कैसे करेंगे. We take this onion. In our practicals we have slide of plant cell and animal cell. Plants I can show here. Now we will remove the peel. What is the peel? This is the transparent portion. See, this is the transparent portion. This is the peel. This is single layered cell. We will keep this onion peel, transparent portion, in the water, and then we will keep this on the slide and see under the microscope, compound microscope, first in the low power, then in the high power. तो जब भी हम स्लाइड बनाएंगे इसको उसके रखेंगे. वी हैव टू पुट कवर स्लिप. कवर स्लिप हमें रखनी पड़ती है ताकि एयर बबल्स नहीं आएंगे. बट दिस यू विल डू इन द प्रैक्टिकली. एंड एनिमल सेल के लिए हम यूज करेंगे चीक सेल्स. And so this completes your chapter, and in this chapter there are this activity, but osmosis is this you can easily do at home, and 2 practicals to see plant cells and animal cells from the cheek cells and the onion peel we will do in the labs. Thank you so much.